Hello everybody, it's your friend Daniel here and welcome to another past paper video. Today we're going to be covering AQA GCSE Biology higher tier paper 1, and we're going to be covering the specimen paper, which means this is once again a paper which nobody actually sat, but AQA just created so people could get a better idea of what their exams were going to be like. Now, jumping straight into question 1. Plants transport water and mineral ions from the roots to the leaves. Plants move mineral ions from a low concentration in the soil to a high concentration in the root cell. What process do plants use to move these mineral ions into the root cells? Now what you have to notice here, it says from a low concentration in the soil to a high concentration in the root cell. Now if it was from high to low, that would be either diffusion or osmosis. Because it's from low to high, that means it's active transport. And question 1.2, describe how water moves from the roots to the leaves. Now, water is moved from via transpiration stream up from the roots to the leaves through the xylem. This is meant to say stream. No, it is meant to say stream. My bad. And yeah, just water is moved via transpiration to the leaves through the xylem via transpiration. Now here we're given a bit more additional information. They like to do this quite often. Plants lose water through the stomata in their leaves. The epidermis can be peeled from a leaf. The stomata can be seen using a light microscope. And we're just showing some data of the amount of stomata in a particular area for the upper surface and the lower surface. Now, 1.3, we're just meant to describe how the student may have collected the data in Table 1. It's a nice free marker. The student would mount the collected upper epidermis on the slide and view under a light microscope, counting the amount of stomata in one area before repeating in 4 other areas and calculating the mean. Before repeating this process for the lower surface and once again calculating the mean. Now, you could do you didn't need to state upper and lower epidermis. You could just say do it for one side of the epidermis then the other side, but I thought just to give a bit more clarity to state one epidermis specifically and then state the other by name. And what is the median number of stomata on the upper surface of the leaf for question 1.4? Now, if you can see from here, you take this list and then you just write it out in order, 0, 1, 1, 3, 5, and take the middle number to calculate the median, which is 1. And then question 1.5, calculate the value of x in table 1, giving your answer to 2 significant figures. Now, to calculate a mean, it's just the sum of all values divided by the amount of values, so you just get the values from up here, add them all together, divide by 5 to get 41.2. Because it says to give your answer to 2 significant figures, you need to make sure to do that and then give it as 41. Now for question 1.6. The plant used in this investigation has very few stomata on the upper surface of the leaf. Explain why this is an advantage to the plant. Now what you need to think about the upper surface of a leaf is there's going to be a lot more sunlight on it. So as the upper surface of the leaf is exposed more so to the sun, with the few amount of stomata on the upper surface, meaning less water is lost, so the leaf will wilt. So there are more stomata on the upper surface, the sun would sort of evaporate water from it, which would cause the leaf to wilt. And with the stomata being on the lower surface, which is more so protected from the sun, this won't happen. Moving on. Question 2. The tobacco mosaic virus is a disease affecting plants, and we're just shown a nice figure of it, where yellow patches where TMV has destroyed chloroplasts, which is like black and white. I don't know why AQA is expecting us to work with colour when it's black and white. All tools should be washed in disinfectant after using them on plants infected with TMV. Suggest why. Now this is to prevent the virus from spreading to other plants. Now if you think it through, if you use like hedge trimmers on a hedge that has TMV and then use that hedge trimmers on a healthy hedge, the virus will then infect the healthy hedge, meaning it's unsafe to use tools that have been used on plants infected with the virus on other plants. Now for 2.2, scientists produced a single plant that contained a TMV resistant gene. Suggest how scientists can use this plant to produce many plants with the TMV resistant gene. Now what you can do is you can take cuttings or stem cells from the meristem to create more plants with the resistance gene. And this can be done by just taking cuttings and then connecting them to other plants, or just planting the cuttings, so that all future plants will contain this gene that prevents them from being infected with TMV. 2.3. Some plants produce fruit which contain glucose. Describe how you would test for the presence of glucose in fruit. Now, this is always a big topic, food tests in biology. Usually it's either a 6 marker, which appears quite common, and for this particular paper, there's a 2 marker here, and then there's another 2 marker later on. Now, you just need to remember what it is, and for glucose, you need a heat mixture that includes part of the fruit and add a few drops of Benedict solution, and if glucose is present, the Benedict solution would turn from blue to orange. Now, TMV can cause plants to produce less chlorophyll. This causes leaf discoloration. Explain why plants with TMV have stunted growth. The lack of chlorophyll leads to less photosynthesis. This leads to less glucose being made, so less energy is released for growth. This is because glucose is needed for respiration, and also there is less protein for growth because glucose is needed to make these proteins. Fairly 4 markers, but this contains all the marking points you'll need to get the full marks on this question. You just need to be a bit careful saying why the glucose is important for growth and why lack of glucose prevents growth. Okay, on to question 3. Microorganisms cause infections. The human body has many ways of defending itself against microorganisms. Describe 2 ways the body prevents the entry of microorganisms. Firstly, stomach acid kills pathogens in food, like if you eat something which contains bacteria, a lot of the time the stomach acid will just kill it, preventing you from getting ill. However, this obviously isn't 100% effective, as anyone who's ever had food poisoning will tell you. And also the skin forms a barrier, so if you have bacteria on your hands or on your arm, it's not going to be able to get into the skin and infect you. It will only do that if you breathe it in, or if you don't wash your hands and then use your hands to eat food. Moving on. 3.2. So it's a nice 6 marker. In 2014, the Ebola virus killed almost 8,000 people in Africa. Drug companies have developed a new drug to treat Ebola. Explain what testing must be done before this new drug can be used to treat people. Now, this is, in my own personal opinion, a rather hard question, because drug testing is a fairly niche topic, and it usually just comes under the scientific experiment part in peer review and stuff like that. But I'm just going to read out the question and then explain a couple of parts of it. We can guess preclinical trials of the new drugs on cells or animals to test toxicity, dosage, and efficacy. And after this, you can move on to human testing on healthy volunteers at low dosages, so you can see how safe it is and look for side effects. And then after proving its safety, do trials with higher dosage and check for efficacy. Then you can do a double-blind trial with placebo, which doesn't contain the new drug, and randomly allocate patients to groups, so neither the patients or the doctors knows who is taking the real drug and who has the placebo. And then the trials will need to be peer-reviewed to help prevent false claims and remove bias. First thing first, efficacy, it's just a fancy word for how good it is. And the main 3 stages are you just want to do preclinical trials on things that aren't humans, to check the drug actually works at treating Ebola. And then you want to check it's safe on humans. And then you want to check it's efficient on humans. So it's a sort of 3-step procedure, with the first one being just it's good against Ebola in general. Secondly, it is checking how safe it is on humans. Then on just a higher clinical trial, and then a double-blind test to check it actually works for humans, and it being peer-reviewed to just make sure it's reproducible and it's actually good at treating Ebola. As I said, it's a fairly hard 6 marker, fairly content intensive, but you just need to sort of remember the order of things and then just start writing and hope it comes to you. all the other contents related to preclinical trials. Now moving on to question 4. All living cells respire. That's, I think, the shortest preamble I've ever seen to a question. Just 4 words. Usually it's a lot longer and has diagrams and stuff. Now, 4.1. Respiration transfers energy from glucose for muscle contractions. Describe how glucose from the small intestine is moved into the muscle cell. Glucose is absorbed via diffusion into the bloodstream, and the blood delivers glucose to the muscles via the capillary. So it goes into the bloodstream in the small intestine, gets transported through arteries to muscles, and then it goes through the capillaries at those muscles into the actual muscle cells. Now, this is the usual preamble I see. Figure 2 shows an experiment to investigate anaerobic respiration in yeast cells, and you're just showing a little diagram of what that experiment would look like. What is the purpose of liquid paraffin in tube A? Now, the whole purpose of this is that you want to look at anaerobic respiration, which means without oxygen. So what the liquid paraffin does is it prevents oxygen from reaching the yeast cells. So you just give them a little multiple choice. It doesn't help prevent evaporation. It wouldn't help stop the temperature getting up. It would theoretically help water getting in, but like that wouldn't affect the anaerobic respiration. So the answer is to stop air getting in. And you're once again given the indicator of tube 2 shows changes in the concentration of carbon dioxide. The indicator is blue when the concentration of CO2 is very low, green when the concentration of CO2 is low, and yellow when the concentration of CO2 is high. Now, what colour would you expect the indicator to be in tube 2 during maximum rate of anaerobic respiration? Now, you would expect it to be yellow because when anaerobic respiration is happening, it produces a lot of CO2 and there's no oxygen present. Now, moving on. 4.4 suggests how the experiment could be changed to give a reproducible ray to measure the rate of reaction. And you could collect the produced CO2 in a gas syringe over a certain period of time using a stopwatch or a timer. Okay, moving on. 4.5. Compare anaerobic respiration in a yeast cell with anaerobic respiration in a muscle cell. Yeast produces ethanol when aerobically respirating, but muscles produce lactic acid. Yeast produces CO2, but muscles do not. And both produce relatively small amounts of energy. And that's why it's always best. That's why, if you know, for like humans, aerobic respiration produces a lot more energy. And that's why it's often used for events, why it's often used for like sporting people who have a lot of events that take a while and need a large amount of energy, like marathon running. Anyway, moving on. Question 5. This is the sort of preamble I'm used to in questions. A student investigated the effect of different sugar solutions on potato tissue. This is the method used. It's just a method. If you've done required practicals, you know what it is. Quite often they have a 6 marker where you need to repeat this method. And here you're just shown the results. And here you're just expected to calculate the percentage change. Now, the percentage change is change over original times 100, which is 0.15 over 1.35 times 100, and that equals 11.1%. And this probably should be 11.1% because everything's given to 3 significant figures, so you should probably match that. Yeah, 11.1%. Now, why did your student calculate the percentage change in mass as well as the change in grams? And that's because each potato cylinder had a different starting mass. So if you have like an absolutely giant potato cylinder, or just like a regular potato, it's going to lose a lot more mass. However, the percentage change wouldn't actually be that different. So it's the percentage change that matters, not the actual change in weight. And then we're just expected complete figure 3 using the data from table 2. And then we're just supposed to put percentage change against concentration of sugar solution. And obviously, I've plotted all the points correctly. Because I'm doing this on the PDF, I don't have access to a ruler, but obviously in the exam use a ruler. And just draw all the points, do a line of best fit that sort of separates them evenly. So it's going through 3 points and then there's 2 points on either side that's like slightly different, same distance apart. And then on the bottom you have a scale going from 0 to 1. And then we're meant to use our graph to estimate the concentration of the solution inside the potato cells. Now, when the concentration of solution is the same as the concentration inside the potato cells, there isn't going to be any osmosis of water in or out of the potato, which means there wouldn't be any change in mass, which means the concentration inside the potato cell is where it meets the x-axis, which would be 0.5. And as you can see, that's what I wrote. Now, for 5.5, the results in table 2 show the percentage change in mass of the potato cylinders. Explain why the percentage change results are positive and negative. Now, as what I said, as from like 0 to 0.5, water moves from into the cells, because the constant, and from 0.5 upwards, water leaves the cells via osmosis, meaning that the change in mass can both be positive and negative, depending on whether or not water leaves or enters the potato. And suggest 2 possible sources of errors in the method given on page 16, which is this method. And I just went for drying off the potato cylinders. If you don't correctly dry them off, they're going to weigh more because the water and the sugar solution left on the outside of the potatoes. And potential 0 errors in the scale. So like if your scale is reading as 0 when it is reading as greater than 0 and there's nothing on it, that would lead to errors because the actual what you weigh the potatoes to be, it will be more than their actual weight. Okay, moving on. Question 6. Figure 4 shows the human digestive system, and there may be some slight hints to what the next question is going to be based on the arrows that I have drawn. But the next question is, label the stomach and the pancreas. In most diagrams, the stomach is the thing that leads to the mouth and the trachea, and the pancreas is the thing laying between the stomach and the intestines. Okay, bit more preamble. Many people suffer from stomach ulcers caused by a species of bacteria called... I'm not even going to bother to attempt that. The stomach is lined with a protective lining of mucus. This bacteria is acid tolerant, which can damage this mucus lining. Suggest how an infection with the bacteria might result in a stomach ulcer developing. The bacteria isn't killed by acid, so they damage the mucus lining of the stomach. I was meant to say stomach. So the acid damages the stomach tissue, causing an ulcer. So basically the bacteria survives in the acid, damaging the mucus lining, and then the stomach acid can reach the actual tissue in the stomach, causing significant damage and an ulcer. This bacteria can also cause stomach cancer. Describe how a person infected with it could also develop liver cancer. So if the stomach cancer is malignant, it can spread to other organs such as the liver via the blood by forming a secondary tumour. So basically the cancer spreads, a bit of it can break off into the bloodstream if the cancer is malignant, and then that bit of the cancer attaches to a part in the liver, causing liver cancer. Okay, just moving on to question 6.4. Gluten is a form of protein found in some grains. Describe the test you would use to find out if protein is present in food. Now this is the same part as earlier, just another food test question, is just another thing you need to remember. And for protein, it's burette agent. Add burette agent to the food sample, and a purple colour will be present if protein is present in the sample. And now it's just the celiac disease, with this being a 4 marker. Celiac disease is a disease of the digestive system. It damages the lining of the small intestine when foods that contain gluten are eaten. When people with celiac disease eat foods that contain gluten, their immune system forms antibodies to gluten. These antibodies attack the lining of the small intestine. This causes inflammation in the intestines and damages the villi. Symptoms of celiac disease include poor growth, suggest why a person with celiac disease might have this symptom. So basically, this part is useless, and you need to think about how the actual symptoms, which is inflation in the intestines and damage to the villi, would cause celiac disease. Now what the mark scheme goes off is that damaged villi reduces the surface area of the villi, which decreases the absorption of food molecules by preventing them from diffusing into bloodstream. Therefore, fewer glucose and amino acids will be absorbed, with less glucose leading to less energy for respiration, and fewer amino acids, meaning there is less building material for proteins, both of which stunt growth. So it's a very nice cause and effect question where you're given symptoms and then you need to describe how those symptoms cause the poor growth. A gardener is looking at the plants in his greenhouse. Once again, a rather short preamble. Some of the plants have a disease. Give 2 ways the gardener could identify the pathogen infecting the plants. And it very clearly shows that this is AQA's first paper for this specification, with it being the specimen paper, because I think these answers are dumb and stupid. One is just compare to pictures in gardening manuals / websites, and looking at their mark scheme guidance, which is very positive, they'd probably accept just Googling the symptoms, or like nowadays, like using Google Lens or something to work out what is wrong with the plant. It's a dumb question. And the other mark scheme point is also dumb. Send a sample to the laboratory for testing. Like, it does say it's specifically a gardener looking at plants in his greenhouse. However, most people are just going to be thinking of their general garden and stuff, and this isn't available for most people. Most average people with a garden aren't just going to get a sample of their plants and send it to a laboratory for testing. There may be some like very crazy over the top gardeners out there, but I don't think AQA should be targeting their mark scheme at crazy over the top gardeners instead of standard people. 7.2. Plants can become unhealthy if they do not have essential mineral ions. Describe the appearance of plants with nitrate deficiency and magnesium deficiency. And once again, this is just something you need to remember. It's in your textbook. They rarely ask questions on it, but when they do, if you remember, it's just 2-3 marks. Nitrate deficiency leads to stunted growth. Magnesium deficiency leads to the yellowing of leaves, which is also known as chlorosis. Now, a bit more amble for 7.3. Plants need other mineral ions. Potassium ions are needed for healthy root growth. Phosphate ions are needed for healthy flowers and fruits. The gardener makes his own garden compost. The percentage of minerals in his compost was compared with 2 fertilizers he could buy. And here we're just given a little table of their cost and the amount of nitrate, phosphate, and potassium ions. The gardener chooses to buy fertilizer S. Explain why you chose fertilizer S. Fertilizer S has the highest nitrate ions and has the highest potassium ions. It is also cheaper than the other fertilizer. But the 4th mark scheme point is a bit weird. It has less phosphate ions, so flowers and fruits are perhaps less important. Now, for this, you need to read the question carefully, where it says phosphate ions are needed for healthy flowers and fruits. And because this guy's a gardener, he probably knows what he's doing, so he probably chose the one that had low phosphate ions for a reason. which means that flowers and fruit probably aren't as important to him. It's a bit tricky, but you just need to sort of read through the question carefully and you'll use a bit of critical thinking. Now for question 8. Lungworm is an infection. Lungworm can kill dogs. Unfortunately, it is caused by a small worm. Figure 5 shows the life cycle of the lungworm. Lungworm reproduces in the dog and infects the lungs. The young stages are coughed up from the lungs and swallowed. Worm larvae pass out in feces and are picked up by the snails, and then the dogs touch or eat the snails, infecting them. What type of organism is represented by the snail in the life cycle of the worm? Now, it's not a fungus, because obviously a fungus isn't a snail. It is a vector, because the vector is the thing that is carrying, sort of like the parasite, and carrying the organism and helping it pass on, like how mosquitoes are vectors for malaria. The snail takes out the same role for the lungworm. Now, if it was asking what was the lungworm, it would be the parasite. And I've had a couple students get that confused before and just not read the question carefully and think they were talking about the lungworm and just instantly tick off parasite, which is wrong. 8.2 suggests how the spread of lungworm disease can be prevented. You can isolate infected dogs to prevent them from spreading it to other dogs. You can treat infected dogs. There's a lot of medicine for dogs for like lungworms and other certain parasites like ticks and stuff. And you can destroy the snails. Destroy the snails is word for word from the mark scheme. There were other ones that were like slightly more obvious, but I just think destroy the snails is a very simple message and kind of a bit funny that AQA has included it just in that form instead of saying like destroy the infected snails. It's just destroy the snails with no context, which I just find a bit funny. 8.3. Malaria is a disease spread by mosquitoes. Describe 2 ways to control the spread of malaria. You could stop mosquitoes from reproducing. Like, I know there's a lot of schemes going on around the world where they release infertile mosquitoes into the wild, which completely just cuts down the population of mosquitoes. And use mosquito nets, which just you put around your bed to prevent mosquitoes from getting in, being able to suck your blood and infect you with malaria. Surprisingly, despite destroy the snails being a valid answer, you don't get any marks for just saying to kill the mosquitoes, which I think's personally a bit weird. Now, moving on to question 9. Figure 6 shows photographs of some animal cells at different stages during the cell cycle, and you're just shown figure A, figure B, C, and just figure A, B, and C. Now, which photograph in figure 6 shows a cell that is not going through mitosis? Now, A is obviously undergoing mitosis because the cytoplasm and cell membrane are splitting apart. B is a bit less obvious, but they're sort of all grouping into the center, all the nuclei and chromosomes, which is part of mitosis. And for figure C, it's just a normal cell. No mitosis going on there. And describe what's happening in photograph A. As I said, the cytoplasm and cell membrane are diffusing, with this leading to the production of 2 identical daughter cells. A student wanted to work out more about the cell cycle. The student made a slide of an onion root tip. She counted the number of cells in each stage of the cell cycle in one field of view. Table 4 shows the results. And it's just number of cells with non-dividing cells and the various stages. Each stage of the cell cycle takes a different amount of time. Which stage in table 4 is the fastest in the cell cycle? Now this is a bit of a tricky question, but what you need to think about is how many are in each stage. So if there's 36 cells and one cell is in stage 4, 1/36 of the time it's in stage 4. So like if you think about it, like throughout the various stages, it spends a ninth of its time. It spends 9/36 of its time in stage 1, 4/36 in stage 2, 2/36 in stage 3 and 1/36 in stage 1. It just requires a bit of maths knowledge to sort of think about it as in like time spent in various stages. And stage 4 is the fastest because only one cell can be seen in this stage. The cell cycle in an onion root tip takes about 16 hours. Calculate the length of time stage 2 lasts in a typical cell. Give your answer to two significant figures. Now, as I said, if you think about it like proportion of time spent, it spends 4/36 of its time spent in stage 2. So what you need to do is you need to times 4/36 to get it in 4/36 times 16 to get the amount of time spent in stage 2 in hours. And then you simply times it by 60 to get it in minutes, and that gets you 107 minutes. However, because it wants it in two significant figures, for your actual answer in the answer box, you need to write it as 110. Otherwise, you would only get 2 of the 3 marks. Bacteria such as, once again, I'm just not going to try to pronounce bacteria names. Coli, let's just E. coli, yeah. Figure 7 shows a growth curve for E. coli growing in a nutrient broth. What type of cell division causes the change in number of E. coli cells at P? This is just binary fission. That's once again something you just need to memorize from your textbook. Not much you can do if you don't remember it. And suggest why the number of cells levels out at Q. And as you can see, it stops increasing at Q. Also, very weird that they've done log graphs for this. Log graphs are only something you really see in A-level sciences. Like, they don't expect you to know anything about log graphs, but they've given you one anyway. And this is due to there may be a shortage of nutrients in the nutrient broths, leading to cells dying out at the same rate as cell division. So basically, there's not enough nutrients for all the cells to survive, so some cells start dying. However, the cells that are dying are being replaced by new cells that duplicate at the same rate, which means it stays constant. Figure 10. Explain how the human circulatory system is adapted to supply oxygen to the tissues and remove waste products from the tissues. This is a fairly content-heavy question, and I'm just going to jump right into my answer and then explain as I go along. It's a lot of words. You won't need all of this to get. There's like I believe 11 or 12 marking points for this question, and if you format your answer correctly, you'll only need like 6 to 8 marking points to get full marks. The human circulatory system is a double circulatory system or a double pump system. I'm writing that, but which means that it has higher blood pressure and a greater flow of blood to the tissues. The human heart is made of specialized muscle cells, which have long protein filaments that can slide past each other to shorten the cell to bring about stronger contractions for pumping blood. Our heart pumps blood to the lungs and pulmonary artery so that oxygen can diffuse into oxygen from air in alveoli into the alveoli blood, and then can diffuse into blood from air in alveoli. Sorry about that. Need a full stop. Need to work on my grammar. And then blood returns to heart via pulmonary vein, where muscles pump blood to the body via the aorta, and oxygen is carried by specialized red blood cells, or RBCs, which contain hemoglobin to bind to oxygen and have no nucleus, so there is more space to carry oxygen. Arteries then carry oxygenated blood to tissues, where capillaries deliver oxygen to cells for respiration and energy release. And the thin walls of the capillary, that one cell thick, allow for easy diffusion to cells, with the large surface area of capillaries to maximize exchange by increasing the amount of diffusion. And then blood goes back into the back to the heart in veins, which have valves to prevent backflow. Cardiac output can vary according to demand and is affected by adrenaline. Now, these questions sort of mean because it's a weird mix. Like, personally, if I was seeing this paper, I wouldn't talk about the actual flow of blood around the body. I would just talk about what makes it more efficient, such as the specialized red blood cells or thin walls allowing for more diffusion. It is a hard question, But even if you're just able to talk about the flow of blood around the body, that should get you 3 marks, as the grade boundaries for combined science, this isn't combined, the grade boundaries for science are usually rather low, which means this is going to be good for that. Anyway, moving on. Question 11. Don't know how that got there. Anyway, monoclonal antibodies are used to measure the level of hormones in the blood. Pregnant women produce the hormone HCG. HCG is excreted in urine. Figure 8 shows 4 pregnancy test strips, and it's just 4 pregnancy tests. Positive test result, a line appears in the control window and the result window. Negative test result, a line only appears in the control window. Invalid test result, no line appears in the control window. It asks which test strip shows a negative test result, and the answer is A, because control window is there and result window is empty. B would be invalid, because no line appears in the control window. C would be positive. D would also be invalid. And give one other use for monoclonal antibodies. To treat cancer. It's not on the mark scheme, but it's also used for sports doping detection. I think that's one of the more interesting uses, but unfortunately it's not on the mark scheme. And then we're just showing the part of a pregnancy test strip, and explain how the pregnancy test strip works to show a positive result. Now, weirdly, this isn't a leveled 6 mark, it's just 6 marks. That's not leveled, so I've just copy and pasted the mark scheme to go through it because it's not leveled. Firstly, urine passes through the reaction zone. The HCG hormone binds to the mobile HCD antibody in the reaction zone. And as it passes up the stick, it binds to the immobilized HCD antibodies in the result zone, which the other antibodies do not attach to. And they bind to the antibodies in the control zone, and blue dye appears in both control zone and the result zone to show a positive result. Anyway, that's the end for this video. Thank you for joining. Make sure to like and subscribe. And yeah, overall, this was a fairly nice paper. Like, it's one of the harder ones because it's specimen, but it's not hard as in content-wise. It just has a couple of weird questions that I wouldn't expect to see in a regular paper. Yeah, anyway, thanks for joining, and I'll see you next time.